Yeah, Immune System Part 1 notes today. Okay, is anybody not done with your data question?
What was the answer?
Let's go over it. Okay, so the question said, basically, what is this picture showing you? It says, the picture shows a white blood cell using its pseudopodial foot to engulf an invading bacteria. First of all, before we go any further, what does, first, it tells you white blood cell, but what is a pseudopodial foot? So, this word right here.
Fake.
Good. Pseudo, my sophomores played the game last year. Pseudo means false. So pseudo is false. Pod is foot. So pseudopod is a false foot. And Eli said it reaches out and grabs something and pulls something in. So if it's a white blood cell, it's going to pull in a bacteria. But if it's like an amoeba, it's going to pull in some food. Right? So it reaches out, and that's how they move. So some small cells don't have cilia, like the little carpet-like things, or flagella to move. They move with a pseudopod. So what it is their cell membrane like reaches out, and then they can like pull themselves towards that. So the word pseudopod means false foot. They can reach out and kind of pull themselves towards it. So it says the picture shows a white blood cell. That was a giveaway right there. Using its pseudopodial foot to engulf an invading bacteria. So somebody tell me I knew it wasn't that answer and why. You knew it wasn't B. How did you know it wasn't B? Okay, what's a plastid? Something in plants, right? It's a thing that helps plants capture energy and make sugar. So it's like in the thylakoids. Not going to be engulfing bacteria. But I'll be honest, that wasn't the one I marked off first. The first two I was able to mark off was A and D.
Because we don't know anything about SCR or ER.
We knew everything about smooth ER and rough ER. Okay, so just as a review, somebody tell me. No, you're fine. You didn't know that SCR was smooth ER?
No, I thought it was like,
that did not even cross
on a
my
test,
mind.
would you put smooth?
I kind of guessed that.
I copied this question, so probably not. Yeah, okay, so somebody tell me, just as a reminder, this is just a review, what does the smooth ER do?
It
Stores calcium, detoxes blood,
it makes carbohydrates and lipids.
it helps breaks down carbohydrates, and it makes lipids. Right, so remember, lipids are made on top of it. That's like its main thing. Lipids are made on top of it. It stores calcium for muscle contraction. It breaks down sugars or breaks down carbohydrates and detoxes blood. But really, both ERs like transport materials inside the cell. So both endoplasmic reticulums transport materials. So, but all of those things we named, all 5 of those functions we named, don't have anything to do with engulfing bacteria. Right? They don't rid the cell of bacteria. Then D, what does somebody tell me? What does the rough ER do? It doesn't make ribosomes. It transports proteins, and it's where ribosomes are attached, right? Ribosomes are attached. That's what makes it look rough. So ribosomes are attached. It is where proteins are made. Why? Because ribosomes make proteins, and ribosomes are on top of it. Where? Okay, ready? Here's your question. Where are ribosomes made?
RNA.
That's what they're made of. But where are they made? In the nucleolus. Good. So ribosomes are made up of our RNA, and they're made in the nucleolus. So good job. Y'all remember that well. Okay, and then we already marked off plastids because that's the thing in plants. So the correct answer should have been A. Somebody tell me what does... I'm sorry, C. Dadgummit. Somebody tell me what does a lysosome do? It breaks down. It cleans, right? So that should have been easy. And if you read your notes last night, it said lysosome on it. So remember that. Here's something to know. Some white blood cells, not all white blood cells, some white blood cells reach out with their pseudopods and they grab a piece of bacteria, or the bacteria, and they engulf it or they swallow it. What would cell eating be called? Phagocytes. Good. Cell eating would be like endocytosis or phagocyte. And once it gets inside it, it mushes it up. It mushes up the bacteria in the lysosome. That's easy to remember. Lysol kills bacteria. The lysosome rids the cell of waste. Okay. What's the question? Was there a question?
It wasn't really a question.
Okay, things you should already know. The first thing on your already should know list is about ligands. Somebody tell me you know what a ligand is.
Signal molecule.
Signal molecule, right? Okay, so the last topic we talked about was the nervous system. And we talked about how nerves talk to nerves. Nerve cells or neurons talk to neurons, right? Okay, well, the next 3 days, it's all about how do immunity cells, how do white blood cells talk to white blood cells. That's what this is all about. I told you we would talk about reception, transduction, response until Christmas. And we're going to talk about all day, today and tomorrow, how white blood cells talk to white blood cells. They communicate via ligand. You just have to know the certain cells. Okay, and then this picture, the second thing on your should know list, glycolipids and glycoproteins. Somebody tell me, what is the structure of a glycolipid? That's not a hard question. Come on. What's the structure of a glycolipid? Okay, a sugar and a lipid attached to each other on the cell membrane, right? It's a sugar and a lipid attached. So a glycolipid would be right here. That green thing is the sugar, carbohydrate, attached to a lipid. Do y'all see that? Okay, a glycoprotein, what would be the structure of a glycoprotein?
Sugar attached to a protein.
There we go. It's sugar attached to a protein, like right there. Okay, so we got the structure. Somebody tell me, what's the function?
Help communicate.
Cell communication, cell recognition. Okay, imagine this. This is about to be inappropriate and crude. Your cells are blind, and they don't speak.
Helen Keller.
That's not sweet. Okay. That's so horrible, and that is on my video. Okay, so cells, they don't speak with words. They can't see. Your cells don't have eyeballs, in case you were wondering. So they feel, right? They feel with glycolipids and glycoproteins. And like, oh, that feels okay. That's weird. Right? So your cells feel, my arms are sugars, my body is a protein. So I am a glycoprotein. Got it? So that's one way some of you, that when they have to touch, cell-to-cell communication. What would be another word besides communication? Cell-to-cell
Talking.
recognition. I recognize that this is a normal student in my classroom. She feels normal. Right? Okay, better. I recognize that this is my cell. This is a normal body cell. It can stay. I recognize that this is not normal. I need to eat it. Right? Because I'm a macrophage. I'm a phagocyte. So this, like, they feel, they're like, oh, normal. Not normal. Me?
Yeah.
Okay, so the next thing, thank you. It's okay. I thought this was interesting, so I put it up here. Influenza, the flu, in 1918 and 1919 killed 22 million people in only 18 months. So I just thought these were some interesting facts. The next thing it says, 3 million people will die from malaria every year, this year. 3 million people. Do y'all know what malaria is? You know what the flu is. Do you know what malaria is? Malaria is it's a protozoan. Y'all remember from 9th grade biology, there was like animal, plant, fungi, protist, and bacteria? Protist is a protozoan is a protist. So it's in the protist kingdom, and it's like a parasite, and you can only get it from getting bit by the Anopheles mosquito. It's like a certain mosquito that carries malaria, and the mosquito transfers malaria from person to person, but only through the mosquito. Right? So like the mosquito bites me, and then the mosquito would come over here and bite Julia, and now she gets malaria. She got it from me, but only through the mosquito. So we all went to Africa on a field trip, and we all got malaria. Could we come back into the United States?
No.
Yes.
Yeah.
Unless you're planning on bringing a cage of Anopheles mosquitoes, the United States is not in any harm. And that's one reason why they're very cautious letting people, if you ever go out of the country and you come back into the country, they're very cautious. They like check your bags and make sure that you didn't bring any boxes of mosquitoes. Right? Or fresh fruit or fresh flowers that may have mosquitoes in them. Right? You can't bring homemade foods over the border unless they're packed in your suitcase. Anyway, so malaria, and it gives you you get chills and fever, real high fever. It would kill lots of young people. It kills babies most of the time. And older people, things like that. That's right. People with sickle cell are resistant to malaria. Because the protozoan doesn't like the sickle-shaped blood. It doesn't.
Well, if they're sick, won't they inject sickle cells?
That wouldn't work. There's a vaccination for it that lasts about 6 months. Vaccination does, but it's expensive. And so everyone in Africa can't get vaccinated every 6 months. Okay, next thing. Oh, since 1980, 1,900 people have died from AIDS. So 619,000 people. A lot of people have died from AIDS, and you know AIDS is a virus. Okay, so first thing, just a little thought. Do y'all know what an amoeba is? This is not on your neck, so I'm just talking. Do y'all know what an amoeba is?
Amoeba's changed, so...
They change forms because they have pseudopods.
Yeah.
Right? Amoeba is a little, it's a unicellular organism, and if you ever drink it, it makes you really sick. It makes you have amoebic dysentery. What does that mean?
Diarrhea.
Amoebic caused you to have bad intestines, right? Y'all hear amoebic dysentery would be like an amoeba caused you to have bad intestines. Okay, so, but amoebas, they have pseudopods and they engulf food. So that was the first organisms that we knew of that were able to use their pseudopods for phagocytosis. Y'all know what phagocytosis means? Or it can either be pronounced phagocytosis or phagocytosis. Phag means eat. Cyt means cell. So phagocytosis would be eating of a cell. So amoebas were the first cells that we know of to be able to reach out and grab food and eat it. Okay, so check out this question. See what you think. We'll just talk about it. Propose a cell communication mechanism that ancestral amoeba may have used to distinguish between foreign cells and other amoeba cells. Somebody tell me what you think.
Same texture, same protein. They can sense the proteins that were on there.
Okay, so Eli said they can sense the protein on... The question is, how can they distinguish between foreign cells, like food, and themselves, amoeba cells? Like, how can they distinguish between that's an amoeba. I'm not going to eat an amoeba because I'm an amoeba. Oh, and that is a piece of food or another cell, so I'm going to eat that one. How can they differentiate? Eli, say what you said again.
So they pick up the same, like they all have the same protein, DNA or protein on them, and they know that that's like, don't eat that because that's part of the body.
Okay, so he said it pretty simply. Other amoebas have like a certain protein. A surface protein is called an antigen. Okay, so other amoebas have a specific antigen. Okay, now finish that with some more science terminology.
What's the antigen?
No, I got antigen. So some amoebas have a certain antigen, and the other amoebas...
Other amoebas have the same antigen, and they can sense that antigen.
Feel?
They can feel with their foot.
No.
So,
They can, he said feel, they can sense, they can, come on, science words, let's go.
With
What did I just do?
their fake foot.
No, they don't have noses, they can't smell.
Thank you.
Do what? They don't have dendrites.
They use their glycoprotein.
Your dendrites things like
Okay, say that again.
if they get a signal on, I think they're supposed to eat, they'll know that it's not a normal one.
Okay, I like that one better. I like that answer too, but let's finish Eli's answer, then we'll go with your
What did
theory.
he say?
He said that they have certain antigens, and they can feel or sense another amoeba. Instead of feel or sense, that's the word I'm hung up on. Amoebas can
Touch. No.
identify with their glycoprotein. They can identify those surface antigens with their glycoproteins, and they can recognize to eat or not to eat. Got it?
What if they don't?
Okay, so that's just one theory. That's just a theory. Y'all understand that that's all though?
Yeah.
That's just one guess. Okay, now let's go with what Rachel said. Rachel, say that again.
If they received a signaling molecule from a cell that was not amoeba, they would know that they're supposed to keep happening.
Okay, so they communicate. It's just another theory. All we're doing is thinking of one. They not propose, they release a signaling molecule like a ligand. So let's go with what if amoebas release a ligand that says, hey, I'm an amoeba. So don't eat me. We're the same. Don't eat me, we're the same, right? So certain amoebas release a signal molecule so that they can be identified or notified. Does that make sense? Yes? So the glycoproteins would help in identification there.
Oh, well.
Okay, so the first thing on your notes that we're talking about today is innate immunity. That's the immunity that you were born with. I know you've all heard the story that, like, if a woman breastfeeds, then the baby will get the mom's immunity. Have y'all heard that?
Yeah.
That's why, okay, breastfeeding is supposed, now I've never had a baby or breastfeeding, I don't know. But I do know that people who breastfeed do it because while you're breastfeeding, the baby has the mom's immunity. So there's two types of immunity. There's the immunity that you're born with, that all humans have, and then there's your immunity that you have acquired over the years. Right? So I've been working on my immune system for as old as I am. I've built it up nicely. A little bitty teeny tiny baby hasn't built up an acquired immunity yet. Right? They haven't built up any acquired immunity. So today, Tamara is acquired. Today, all we're talking about is innate immunity. The immunity that you're born with, not from breast milk, but what you're born with. So one thing you want to highlight, it says innate immunity, you want to know that this is non-specific. Okay, example. A baby that's like 2 days old. Are they immune to the chickenpox? No. Am I immune to the chickenpox? Yeah, you couldn't start school until you were. Now I don't think. Who in here had the chickenpox? Hands. Only 2 people? Okay, the rest of you, there's only a couple of you. The rest of you had the chickenpox vaccination. So today, sitting here, none of us can get the chickenpox. I don't think. Unless we have something.
You can't get it twice, I think.
Yeah, you won't get it twice, right? Because I have a built-up immunity. I had the chickenpox, like full-blown itchy spots everywhere. So that would be part of my acquired immunity. I wasn't born with the immunity against chickenpox. Does that make sense? Okay, so innate immunity is non-specific, and this is what highlight where it says born with. Okay, it helps protect against most organisms in the environment which are hazardous. Okay, one thing I want you to know. Most bacteria are good for you. Most bacteria are not bad, and quite frankly, they're good for you. Only about 1% of the bacteria on planet Earth is bad for us. So the other 99% is either not harmful or good for us. It doesn't affect us at all. Okay, so a word that you do want to know that's not in your notes, so write this in. Pathogen. Is any organism that makes you sick or any organism that causes a disease. So write that word in somewhere. Any organism that does cause a disease. So today, tomorrow, and Friday are all about how do you fight off a disease. Okay, so my first line of defense, if I come right here and I touch this table, and this table has Streptococcus pyogenes, that's what causes strep throat on it right here. I touch it. Why do I not get strep throat? Because I have my skin, right? Okay, imagine how infected you would be for just 1 second if you didn't have any skin. Well, you would look weird, that's for sure. And you would not have any friends. But you would be so infected, it'd be like oozing infected, right? When you have a big sore on your arm, what are you worried about?
Infection.
Infection. You're worried about it getting infected. So although we don't consider our skin as a part of the immune system, it's like the number one thing we've got against nothing specific, but against everything. All the organisms on your skin right now are not in your bloodstream because of your skin, because they're on the outside. So your skin is a protective barrier that doesn't allow pathogens inside your bloodstream. Got it? So skin, if you're ever asked something about the non-specific or innate immunity, your answer should be skin, if you get to write about it. Okay, so skin and mucous membranes, these are considered the first line of defense. Mucous membranes would be like the earwax in your ears. Bacteria gets caught in there and doesn't get to go into your brain. That's a good thing. Your snot. You breathe it in, you got mucus all up in your snot. That's preventing the bacteria from getting any further. It's a mucus barrier. So it's helping the pathogen. What's a pathogen?
Anything that can cause infection or sickness.
Cause an infection. Good. And cause an infection or sickness, stay out. So the mucus, okay, your eyes, that's not next. Your eyes have like a little lysozyme inside them. Your eyes have a little antibiotic in them naturally called lysozyme. That's also the enzyme inside the lysosomes that help prevent bacteria from getting in your eyes to your body. Now, some things still do.
Is that why your eyes are always wet?
Yeah,
Still one of the
well, that's one of the reasons. But it's just a barrier. I think that eyelashes are to keep sweat out of your eyes, but I'm not sure. But I think that's their purpose. I know that's your purpose of the eyebrows.
Sweat and dust.
Sweat and dust.
But then your
Yeah.
eyelashes
You know when
to
you
your
walk
eye.
through like something that has like a lot of like you can just see everything in the air and it just doesn't catch on your eyelashes.
Oh, okay,
You know there's
so
mites on your
yes.
eyelashes?
I've seen those on videos. Okay, so it says on C, sweat has a pH of 3.5. So something just with a high pH. So sweat, 3 to 5. I don't know. I think I said that wrong. Anywhere from 3 to 5. Well, you can just, look at it. Okay, D is a big one. Your normal flora. Okay, so normal flora is the normal bacteria on your skin that isn't making you sick. So it's non-pathogenic. Normal flora. Okay, so let's look at my hands right now. This is about to gross you out. My hands and your cell phone and your keys and your pencil and your highlighter and the outside of your clothes covered in bacteria right now. On every small centimeter of your skin, there's bacteria. Every small centimeter. Because it's there and it's taking up room, which is a good thing, and that's your normal flora. It's not making you sick. Every time you touch your mouth to your hands, you don't get sick, do you? No, we'd be sick all the time because we all touch our mouths. You have normal flora, which is good bacteria, all over you. And what it does is it prevents the other bacteria from getting on your skin and culturing. Do y'all know what the word culturing means?
Growing.
Yeah, like growing, like making home, making lots of progenies of them. How do bacteria divide? Make me proud. Binary fission. Good. Bacteria divide by binary fission. So this morning I took a shower before I came to school. Yay me. I hope you did too. And I washed my body, my hands and all that. So my hands were pretty clean. And then about right before lunch, I washed my hands again. Now, what was left on my hands after I washed my hands?
Bacteria.
Soap.
No, I washed the soap off, so the soap's gone.
Water.
Some bacteria is left, right? I didn't wash my hands in battery acid. I still got some bacteria on there. The soap doesn't kill all the bacteria. So what kind of bacteria is left on my hands after I washed them with soap?
Good bacteria.
Some good,
Normal
some bad,
flora,
maybe,
normal flora.
right? That's not what I'm looking for, good or bad. The bacteria that wasn't killed by the soap is what's left, right? So I washed my hands with soap, antibacterial soap. So I washed them real good. I rinsed them off. I dried them. So what was left on my hands was the bacteria that wasn't killed by the soap. Yes, so that's easy. And we call that, the word for that is resistant. The bacteria that was left on my hands is the bacteria resistant to the soap. Some good, some bad, whatever. Okay, well then, let's say at 4 o'clock today, I washed my hands with that soap again. What do we kill then?
The one that was left before.
The resistant's not going to be killed by that soap. The ones that can be killed by that soap, right? So let's say at lunch when I washed my hands And I got a lot of the bacteria off, but there's still some left. Well, now that I'm walking through the day, going about my day, those bacteria that are left are dividing. So the resistant ones are dividing on my hands, dividing, binary fission, divide. So when you wash your hands at 3 o'clock, not as much is killed. Why?
Because you have more resistance.
Because pretty much everything on my hands is what's resistant to the soap. Does that make sense? Does it really? Okay, so the more we use soap, the less it's going to work. I'm not saying that you shouldn't wash your hands. I'm just saying it doesn't do much good. Anyway, so that was normal flora. Look at E. You want to see where it says saliva, tears, and pepsin? That all has to do with the enzyme lysozyme. So saliva, that's in your mouth. Tears, in your tear ducts. And then pepsin is an acid in your stomach. But it all contains highlight lysozyme. That's the enzyme that kills bacteria that's in lysozymes. Yes, ma'am?
Why do you cry? What makes you cry?
Well, you're sad.
Sad days.
Yeah, but why would it do that if the purpose is to kill the bacteria?
What?
It doesn't make any
I don't know. That's a really good question.
This makes you sick, you don't cry.
When Drake and Josh stopped coming on.
Probably it's just a release of. I'm just guessing. I don't know.
When Ross and Rachel broke up.
You cried on Friends. No, I would think that the reason you cry is like all those hormones are released, like sad hormones, and that's just like the exterior response to all those hormones in the inside.
It's like getting rid of the hormones.
Is that why your eyes water before you sleep?
Does that why I cry myself to sleep every night?
Yeah.
Anger is a
I'm
hormone.
laughing really hard. Like
That's a good question. I'll have to look it up. Okay, moving on to F. Look with me on F. Important. You want to hear me say this? A phagocyte is a P-H-A-G-E site, C-Y-T-E, phagocyte. This is just going to be the big eater. So here's what you want to know. The cell, the white blood cell, that is innate immunity. Since today is all about innate. What does innate mean? What you're born with, right? Like Lady Gaga, baby, you were born this way? Innate, this is what you're born with. Okay, so the only cell we've mentioned so far is a phagocyte. So you want to know, because tomorrow when I start talking all about these other white blood cells, they're not innate. This one is innate. This one is non-specific phagocyte. What it does is it feels with its glycoproteins. It feels, and then it takes the bacteria in and eats it. Right? So you can see this is a picture right here of a white blood cell. So the white blood cell is this part right here. Engulfing, that's a yeast cell.
It's not strong.
Thank you. My drawing?
Yeah.
The white blood cell is, do you see how it's like covering up the yellow yeast? It's like engulfing it, taking it in. What's going to happen once it gets it in? It's going to dissolve it. It's going to break it down with the lysosome. Okay, so it says on phagocytes, these are white blood cells, you want to know that eat pathogens. What's a pathogen?
Anything that causes sickness or bacteria.
Good, and this is your second line of defense. So if something gets past my skin, past my mucous membranes, into my bloodstream, these phagocytes are inside your bloodstream. White blood cells. Now it's up to my white blood cells to engulf it and chew it up. Does that always work? No, sometimes you got to go to the doctor and get an antibiotic. So this is your second line of defense, though. It says most phagocytes are neutrophils. What that means is neutrophils are big cells. They're named that way because of the way they're colored under microscopes. But neutrophils are phagocytes, but that's what goes to a site of infection and causes swelling. So neutrophils is just a word for a cell that goes to the site of infection, and it's what causes the swelling.
So swelling's good.
Yeah, swelling is good. And we're going to talk about swelling today. Yes, sir.
Don't know if you have cancer, like your white blood cells are like numbered, like there's a lot of more than usual. So like most people with cancer don't get like sick easy, like strep throat and all that stuff.
I don't know about that. I know that if you take chemotherapy, which is the medicine for cancer, it's going to wipe out white blood cells, and then you would get really sick.
Yeah.
But maybe if you're not on the chemotherapy, that's probably a good hypothesis. I don't know. That's a good thinking. Okay, it says on 2, it says large phagocytes are called monocytes. Here's what you want to know. Phagocytes, monocytes, macrophages. If you see any of those words, they mean big eaters. Macrophages, big eaters. Monocytes. And then it says monocytes when they're in the blood and macrophages when they get to the tissue. Here's how. Monocytes is what we call them when they're traveling around in the blood. When they get to the infected site, we call it a macrophage because it starts eating. So it's a monocyte in the blood and then a macrophage. What would macrophage mean? Big eater. Good. Okay. On phagocytes, move by positive chemotaxis. What that means is, okay, let's say you get cut on your hand right here. A big knife cut or something. A yucky infected cut now. Okay, so the cells right here are going to release a chemical, a ligand, that says that they've been cut. They're releasing a ligand. So the macrophages, how do they know to get to the cut? How do they know to go through the bloodstream to the cut? Because the ligand. It's because the chemical. That chemotaxis, they move towards the chemical being released. So did you ever wonder, like when you take a medication, like when you take Tylenol, how does it know to go to your head and help it? Well, when something's in pain, it releases a chemical. And so the bloodstream carries the blood, macrophages, or that Tylenol that you've just taken, to the chemical that's being released. So chemotaxis is moving towards a chemical. Yes, sir? Uh-huh. Yeah, she didn't have I think she didn't have the brain sensor in her, like she had the neurons and she just didn't feel it. Like she didn't have, the part of her brain wasn't right.
When was
The girl with no pain? Maybe 2010, something like that.
Is she still alive? I thought she was
She was on Oprah. I don't know, that's how I heard about it, she was on Oprah.
So you don't feel physical pain or like mental pain? Like
Physical
she
pain.
Kill somebody, she wouldn't feel it.
I don't think so. She had not when she was on Oprah when I watched it.
I would freak my parents out and
Maybe.
that's my start crying.
Maybe. Okay, so you want to know positive chemotaxis. What does that word mean? Logan, go.
What is the question?
What is positive chemotaxis?
Hold on. Can I have an example?
Yeah. Phagocytes move by positive chemotaxis.
It's the way.
Logan, I just said it!
I'm sorry, I was out of it.
Ryan, go.
They follow the chemical.
They follow the chemical. They move towards the chemical. Okay, and then it says they use their pseudopodial movement. You know what that is, right?
Their foot.
Their false foot. And they eat pathogens. It says eaten pathogens are broken down using lysosomes. Okay, so lysosomes, you know what that is. And then peroxisomes, you want to know, right in. Peroxisomes are little things inside the cell that make peroxide, like hydrogen peroxide.
So just
Oh, that's right. They make, they're little, are they organelles? Really itty bitty organelles, if they're organelles. I'm going to say molecules.
molecules that make peroxide.
Yeah, it's just like a lysosome. It's a small sac like a lysosome, but they make peroxide.
So molecules in a cell that make peroxide?
Yeah. No, they are peroxisomes. They are peroxisomes.
Wait, what?
Okay, and then I put this picture up. I just thought this picture was neat. If this is a picture of your trachea, you have two pipes right here in your throat. The one in the front is the trachea, goes to the lungs. The one in the back is the esophagus, goes to the stomach. Okay, so this is a picture of the inside of the trachea. It says trachea lined with ciliated little hair-like.
Trachea?
Yes, that's the inside.
What, that was like a dirt hill with branches going on.
No. It's the inside of the trachea lined with mucus. So that's the inside lined with mucus. The second thing says the esophagus, which is the back one, leads to the stomach. It has a pH of 1 or 2. So the bacteria aren't going to go down into that stomach because it's so acidic. So this is all innate immunity. The urinary tract, that's like the kidneys and the little urethras that lead to the bladder, have lower pHs. Tear ducts, we said that tears have lysozymes. And then reproductive tract also, like the uterus, also has a low pH. So it's either going to be mucus, like the bacteria would get stuck in it and can't go further. The mucus is like a barrier. Mucus is like a barrier. The pH is going to prevent the bacteria from surviving. Do y'all see how this works now? The skin is like a barrier. Okay, and then I have another picture. This is a picture of a white blood cell right here. So this is the white blood cell, and it's engulfing, these things right here are bacteria. So those little things, how are they engulfing the bacteria? They're using their pseudopods, right? And they're going to break it in. Josh, what's going to happen once they get them inside? They're going to break down the pathogen with their lysosomes. Good. Okay, so this picture, when we're talking about inflammatory response, so move on down to the next part of the notes. Like they squish, they ooze. Like they ooze over. They like with their false foot. Okay, so let's say you get cut right here with something that's covered in bacteria. So those little green dots that you see, those are bacteria. So you get cut. This is just how it all happens. You get cut. Well, your blood is going to pool in that area. That's what causes swelling. So the blood, if you see, it says in the notes that the blood carries to the area. The capillaries dilate. What does dilate mean? Open up, right? So the blood vessels that carry it to, they're actually arteries. The arteries carry the blood to that area, open up. And then the veins that would carry it away close up. They constrict. So the notes say capillaries dilate. That's where the blood is taking it to the area, away from the heart, to the cut. So capillaries dilate and venules constrict to trap blood, swell. We got to get blood to the infection, but not away from the infection. So when we carry the blood that way, we're carrying white blood cells. So you get all the blood there. So swelling, when it get, you know how, have you ever felt swelling? How it's hot.
Yeah.
That heat makes the macrophages go faster. So they eat the bacteria. Okay, sometimes what does that cause? When you got after the swelling's gone down, it's pus, right? And all pus is a buildup of dead white blood cells and then like the leftover bacteria. Yike. What was your question?
If swelling's good, then why after a certain amount of time do you have to go to the doctor because swelling?
Well, okay, swelling is natural. I mean, so is a fever. It's a good thing. It's natural immunity. It's your body trying to get rid of the infection. But if the swelling doesn't go away, like sometimes when, like, let's say you twist your ankle, well, there's no bacteria in there for the white blood cells to eat. But your body identifies that as an infection, so white blood cells get there. And it's like it won't go away. The swelling, so sometimes if the swelling doesn't go away, you have to go to the doctor and get rid of it. Same thing with the fever. Like a fever is good. It's trying to increase your body temperature so that macrophages will go faster and the bacteria can't live in the hot environment. But if your fever gets too high, it's going to cause brain damage.
Is that why you can't get like above 106 or something like that?
Looking at me or are you looking at me?
You?
Why are you looking at me? I didn't do it.
So if your fever gets too high, like above 105, it's going to cause brain damage. So that's why moms take their kids to the doctors.
All the time.
Is that why acne is like just bacteria and white blood cells are there?
Yep. Okay, so it says redness, swelling, edema on 1. And then on 2, you want to know the word histamine. That just means that's the inflammatory response. That's like the redness or the swelling. Histamines. Those are little, you can see them in this picture right here. It says blood clotting elements right there on the purple things. Just little histamines. It's gonna make a swelling. Sir. Yeah. Let's think. What's an antihistamine taken for? It does reduce swelling, but what else? It's for allergies. Okay, so let's say you don't get a bacteria up in your sinus cavities, but you get pollen up in your sinus cavities. Okay, so pollen is going to bring the swelling there, and it's going to make the mucus in your sinus cavities act up. So that's all because of the histamines, the natural inflammatory response trying to get rid of the pollen. Well, the pollen's not going away because the pollen is spring outside, so your allergies are acting up. So you take an antihistamine to get rid of that inflammatory response. Does that make sense? So antihistamines are to calm your allergies, to calm down that swelling in your nasal cavities. So great question. Okay, so A. It is aided by chemokinase. These are chemicals that attract phagocytes. So right here, let's look at these things right here. Those are probably histamines being released so that swelling will occur, and then chemokinase being released so that the phagocytes will come to that area. Remember, cells speak chemicals, so all those chemicals. Neutrophils, remember, are what causes swelling. Neutrophils follow the chemical trail to the site of the infection. Macrophages come in and clean it up, and then that's where it talks about pus. Pus is a combination of dead cells and debris. Isn't that nice? Okay, the next thing, I don't have a picture of this, but progenins, this is going to be fever. This is what causes a fever. Priogens. It says fire proteins. What is it? They're released by the macrophages. What are macrophages? Come on, make me proud.
Big eaters.
They're white blood cells that ingest or eat bacteria, right? Okay, they release a protein. They release a protein that makes you have a fever. So when your macrophages are working, you get a fever. When you have an infection, you get a fever. So they go hand in hand. And I really already told you, fevers are good. Fevers are your body's natural way of getting rid of an infection. If you don't get a fever and you're infected, it may mean that your immune system isn't working like it should be. Bad news. Yes, sir.
It gives you chills.
I think it's when the fever starts to break that gives you chills, isn't it? Oh, yeah, that would be you sweating. But that comes with chills, doesn't it? I don't know. I haven't had a fever in a long time. So
It's like the highest point is when it breaks.
Yeah, so it breaks and then you start sweating.
And then you get cold.
Yeah, and then you get cold. So, but the fever's a good thing unless it gets too high. If it gets too high, it might cause brain damage. So that's a bad thing. Okay. So systemic, those go all over the whole body, make you have a fever. Okay, let's talk about interferons. I don't have a picture of those either. Interferons, it says these are chemicals released. They're ligands. That's all they are. They're chemical messages. Interferons are chemical messages released. Write in the word for me, virus.
So interferons are viruses?
No, interferons are released when a cell is infected with a virus. It says these are chemicals released from damaged, invaded cells that are used to warn off cells of incoming danger. My dad had hepatitis. Have I ever told y'all that?
No.
Hepatitis is a viral disease of the liver. Okay, so he had a virus that infected his liver that wasn't curable. Okay, and he lived with it for a long time. It's a chronic virus. So he had hepatitis, and in order to try to help him get better, the doctors prescribed him a medicine called interferon. So what it was is he was prescribed a medicine that our body already makes. His body made it too. He got more of it, trying to get rid of his virus. So interferons, I always associate with viruses, and they are. Okay, so I got this video for you to show you how an interferon works.
The immune system has several defense mechanisms against viral infection. Here, a virus binds to its target cell. As we saw, it fuses with the host cell and releases its genetic material into the cytoplasm.
Okay, so one thing to note, viruses, that's that blue thing right there. I can make this bigger too. Okay, the virus, which is right there, is inside the cell. Viruses go inside your cells. That's how they infect you. And they're going to release their DNA that's going to go in there. Y'all with me so far? So now this cell right here is infected with a virus.
When the foreign genetic material enters the cell and starts to synthesize viral products, the cell is stimulated to make alpha and beta interferons. These immune system...
Okay, so what just happened? Somebody tell me.
Exploded.
No, it didn't explode. Interferons were released. So the virus got inside the cell. Your cell released those chemicals called interferons. And it said interferons A and B. And we don't have to know that. But the virus got inside. So your cell, listen.
Genetic material enters the cell and starts to synthesize viral products. the cell is stimulated to make alpha and beta interferons.
So your cell got infected. So like my dad's, the liver cells, they got infected. So when the virus was inside the liver cell, the interferon was released. Now what? The interferon are those little orange things. Watch.
Interferons can signal to neighboring cells to directly inhibit viral metabolism.
So what did the interferon do? Come on, science words, let's go. The interferon signaled. What did it do? Look at it. What is that? What is that a picture of? Look right there. What did that interferon do? Oh, come on. It binds to the receptor, which blocks. It's like we all said it, but it binds to the receptor and blocks what? The other viruses from getting in. So right here, that little interferon, which is that little orange dot, the interferon bound to the receptor, reception. And that message said, hey, there's a virus in here, watch out. You're going to get infected by a virus, so watch out. Okay, questions on that? Now read your notes and see if it makes sense again. It says, interferons, these are chemicals released from damaged, invaded cells. Your cell released the interferon to warn other cells of incoming danger. But instead of incoming danger, what would you put? Other viruses, right? Okay, it allows the other cells to increase their defense to interfere with the ability to be attacked. So, hey, I'm infected. Save yourself. That's the chemical. That's what the interferon says. I'm infected. You need to save yourself. Okay, the next part of the notes. Major histocompatibility complexes. Okay, everybody hold up a number 1. Okay, so you have a MHC1. That means that you're not a white blood cell. You're like a lining, you're a tissue in the throat, you're a heart cell, you're a bone cell, you're a red blood cell. You're anything but a white blood cell. Right? That's what you are. You got it? Okay, so let's say this is a virus. I used to have my kids make viruses, but now I don't. She gets infected with a virus. It's inside her. You see her inside? She's really sick now. Ugh, she's bad infected. Like bad with that virus. Bad infected. Okay, so she knows that she's infected, but watch this. If I'm a white blood cell, if she's a normal cell, she feels the same. She feels, with my glycoprotein, she feels the same. Why does she feel the same? The virus is on the inside of her. That's what viruses do. They get inside your cells. Okay, so with her little 1, this is a surface protein called MHC1. She's going to hold up a surface protein called what's the surface protein called? This is an antigen, right? The antigen from the virus. She's going to hold up a small piece of the virus and say, I'm sick. Just kill me. I'm completely infected. This is a piece of what's inside of me. This is a piece of what's inside of me. Just go on and kill me before you all get infected too. So this is almost like I surrender. Just kill me. Suicide right here. Go on and kill me. So that's what the MHC1 does. What is it?
Signals suicide.
It signals I'm sick. This is what's inside of me. Come and either eat me or you watch out because it's coming to get you too. So now that I am a white blood cell with my glycoproteins, she feels different. This is how she's communicating with me. She's holding up, what is this thing? It's a piece of the virus, the antigen. It's a surface protein from the virus. She's holding it up, and now she feels different. So I'm a macrophage, I can eat her. Right? Because I eat. Okay, so do y'all understand what MHC1 does? Kayla, tell me what MHC1 does. Okay, so MHC1 is a surface protein that allows the cell to hold up a piece of the antigen, attach a piece of the antigen, right? It attaches a piece of the antigen so that it can notify the white blood cells that it's sick. Okay, so you can see that here. Now, I am a white blood cell. That's what you are all like. muscles or red blood cells or anything but white blood cells. I'm a white blood cell, and I have a 2. So MHC2s are only found on white blood cells. Do y'all see that in your notes? MHC2s are only found on white blood cells. Okay, so when I come across this virus, what do? I'm a white blood cell. I eat it. Nom, 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 nom. It's gone. I took it inside my lysosome. It's all gone. And I can hold up with my number 2. I can hold up an antigen. So she held it up with her 1. I'm holding it up with my 2. And this says, hey, look at this. You watch out. It's coming for you too. Hey, watch out. Watch out. So it's not notifying for me to be killed because I'm not infected. It's notifying, this notifies other cells of what's coming. So MHC2 molecules, hear me say this, MHC2 molecules, it notifies the other white blood cells, look, I killed this. You go, hey, feel this. Now you go look for that, you kill it too. You feel this. So you go look for that, you go kill it too. It notifies the other white blood cells. You got it? Okay.
I know a good way to remember that.
Go tell me.
Paul Revere has 2 words, and he notified that the British were coming.
Good job. I like it. Okay, so I like it. Okay, Olivia, tell me, what's the difference between MHC2 and MHC1?
MHC1 is on the normal cell, and MHC2 is on the white cells.
Good. MHC1 is on all cells but white blood cells. MHC2 is just on white blood cells. Kayla, tell me about their function. Or Allie, you tell me about their function. MHC2 is to notify other white blood cells. It's like a trophy. This is what I killed. You go kill it too. Got it? Okay, the last thing on the notes is just about plants. It's kind of common sense. What do plants have that protects them? What'd you say? Okay, plants have a cell wall. Absolutely. We don't have a cell wall. Don't you think that it would be harder to get a virus into the cell wall? I think so. And Josh said that, and I was like, hey, that makes sense. Okay, what else? Yeah, Thorns is on the picture. Come on. Well, how do Thorns work? How do they prevent? They don't prevent viruses or bacteria. They prevent what? Like mosquitoes from eating the plant or whatever. People from picking them. If you see a flower and it has thorns, you're like, maybe next time. Pick around it or just don't pick it at all. So the thorns help get away predators, like predator prey, or like a caterpillar that would eat the plant. A bug that would eat the plant may not eat it because of the thorns. Okay, what else do plants have? Think about it.
A bark.
They have a bark, a cork. What'd you say? Some plants have poison, maybe like a bad taste, a poisonous taste. So cork, thorns. What about, have you ever seen like a leaf that has a big, thick, waxy cuticle? Don't you think that would prevent a caterpillar from eating it? So that cuticle, okay, one thing to know. Plants don't have, I know you know this, but it's like when you get to writing, you forget. Plants don't have a bloodstream. They don't have macrophages going around in their blood. So like plants, if they get infected, like let's say one leaf gets infected, they've got to have localized immunity. It's not like a white blood cell from this leaf over here can go over there. That sounds crazy, but if you knew the number of times I've read that the white blood cell comes from one side of the plant to the other side. Plants don't transport white blood cells. They do transport water and sugar, but not white blood cells. So one thing that leaves can do, they can, like if one leaf gets infected, they have the ability to cut that leaf off and live without that leaf. Where like you and I, if our foot gets infected, where's the infection going to go? All over your whole body. Where the leaf can cut that part off and that's localized immunity. They can get rid of that one part. Okay, grab you a sheet of paper, please. Sheet of paper. And if you'll put your notes away. Okay, so in order to get 1 point, you had to identify skin. Describe it. It is a protective barrier that doesn't allow pathogens in. Okay, mucus, you might have talked about that one. Mucus would be your ID. The describe the describe is it's a barrier. It doesn't allow pathogens to come into the body. If you use thorns, you've got to say it's a protection against herbivores. A protection because it's
animals, like protect against animals.
protect against animals because it's prickly. Like you have to say the obvious. You can't just name them or say it protects against pathogens. It does more. How does it protect against pathogens? It is a protective barrier. Like you've got to learn to use your descriptive words. So you get 1 point for ID and describe, 1 point for ID and describe, and then 1 point for ID and describe. So it's 3 points possible. But you only get the point if you identify and describe. You have to identify and describe. And I just thought, hold on to that for a second. I didn't talk about some of this stuff. On this picture right here, it's just showing you like plants have a physical barrier, and then they have metabolites on the inside. And then the third one would be their immune response, like maybe losing the leaf or whatever. And then just a topic that's kind of interesting. It says certain herbivores and plants have co-evolved with one another. This butterfly can break down the secondary metabolite. So this butterfly can break down the poison, and so it's not killed by the poison, and use a nitrogen byproduct in protein metabolism. So I just thought that was interesting how they've co-evolved.